Continuing on with the huge diversity of how ecology and evolution feed into genomics, first we have Karen Kapheim, who will be talking about eusociality in tropical sweat bees. Don't forget to unmute, and then by all means jump right into your presentation.
Okay, good. Can you see my slides okay?
Looks great.
Yep.
Good. And hear me? Great. Okay, well, thank you so much to everyone who's done such a tremendous amount of work organizing this conference. The transition to online has been pretty much seamless as far as I can tell, and I know that's because of all the work you guys have been doing, so thank you so much. Thanks also for the opportunity and invitation to tell you about some of the work that we've been doing with bee genomes recently. So this is basically the question that's been motivating my research for many years now, and that is, how does social behavior evolve? So many in this audience may be aware that there are more than 20,000 species of bees, and the vast majority of them spend their lives at this end of the social continuum as independent replicators, completely solitary. They're essentially single moms making a go of it on their own. But within the bees, there have been several evolutionary transitions to some form of social organization. And among the species with the most complex forms of social organization, they actually can represent what we consider a superorganism, where individuals living within a colony sort of are analogous to individual cells living within a body, some specializing on reproduction and others on maintenance. And so my research has been trying to find answers to how and why these evolutionary transitions occur. And to search for those answers, I've focused on the bee family Halictidae, because within this family, there have been at least 2, but possibly 3 independent origins of eusociality from an otherwise solitary ancestor. And so recently, my collaborators and I have constructed 2 new genomes for 2 species within this group. One is the solitary alkali bee. This is Nomia melanderi. It's an important native pollinator of alfalfa in the western United States. And then the other is this ogochlorine, the facultatively eusocial bee, Megaloptogenalis. So I just want to take a moment to acknowledge all of my collaborators and co-authors who did a lot of work on these 2 genomes. All of the heavy lifting in terms of assembly and annotation was done by Goji Zhang, Hailan Pan, and Sai Li. Rob and his group did the orthology. Eckhart Stoll annotated our transposable elements. We used these genomes in several evolutionary genetics analyses with contributions from Burrell Jones, Brock Harper, Clement Kent, Amra Zayed, and Ariane Avalos. And then these projects were supported in large part by the Smithsonian Tropical Research Institute, particularly Bill Weislow, who pioneered the study of Megaloptogenalis. So these genomes are, despite the fact the alkali bee genome was just published about a year ago, the Megalopta genome was just published less than a month ago, and they are already dinosaurs. So these were the first projects I started in my lab basically 6 years ago to the date. And as happens, we got the assemblies relatively quickly, and then we spent several years studying them, analyzing them to answer our questions about social behavior. So they're a little outdated. They're Illumina-based assemblies. Despite that, they're pretty good as far as bee genomes go, with the N50 scaffolds being several megabases. The contigs, of course, are highly fragmented, but our BUSCO scores tell us that we have most of the genes that we need to answer the types of questions that we're interested in. So another step that we took with these genomes was to discover the microRNAs that are expressed in the brains of these 2 species that I just highlighted, as well as 4 other bee species. The size of these pie charts here corresponds to the number of expressed microRNAs we found in each species. But what's particularly intriguing are that roughly 1/3 of the expressed microRNAs are unique to that species, with, in fact, a large proportion of them, as indicated by these black slices of these pies, having no known homologs in any other insect species. So we wanted to investigate these a little more, figure out what they might be doing in the brains of these bees. So we used computational methods to predict their messenger RNA targets. And so here you're looking at a bunch of pie charts. The size of the pie here corresponds to the number of predicted target genes for those lineage-specific microRNAs. And the color of each slice in the pie is corresponding to the age of the orthogroup to which those predicted target genes belong. So if you'll look carefully, you'll see that the green slice of this pie is relatively large compared to the green slice over here, which corresponds to all of the genes in the genome. And those are the genes that are unique to that species. At least they don't have any known orthologs at this point in time. So what this tells us is that these lineage-specific microRNAs are disproportionately targeting the lineage-specific genes, which suggests the hypothesis that novel microRNAs may be co-evolving with novel genes as sort of built-in regulatory elements. So we discussed this hypothesis and other hypotheses about the evolution of microRNAs in bee genomes in this paper that was just published about a week ago. So check that out for more. But I want to use my remaining time to tell you about insights we gained about how social behavior may be evolving by looking at this Megaloptogenalis genome. Okay, so Megaloptogenalis is actually the bee species that I studied during my PhD many years ago now on Barrow, Colorado Island, situated in the Panama Canal. It's pictured here. And the reason that I'm focused on this bee is because it exhibits this social plasticity. So in this population and throughout its range, you can actually find both solitariness and eusocialness co-occurring in both space and time. And the way this plays out is that when you have a new nest foundress, pictured here, they're twig nesters, so they excavate tunnels in dead sticks. Once they do that, they have a choice. They can follow a social trajectory. If they do that, then in their first brood of offspring, they're producing both sons and daughters. And for the most part, those daughters stick around and become workers. These workers are functionally sterile. They don't mate or lay eggs. They do most of the foraging for the colony. They feed the queen through trophallaxis, and they help to raise their siblings. But not all the females choose this option. About a third of them pursue the solitary trajectory, in which they don't lay any female eggs at all. They have exclusively sons in their first brood of offspring, essentially precluding them from ending up with any workers. Now, Kim, in the last session, introduced us nicely to the fact that Hymenoptera have haplodiploidy sex determination. So you may be wondering, okay, well, are these solitary females just the ones who failed to mate and therefore they can't fertilize any eggs and have daughters? And the answer is no. Basically, every solitary female we've looked at has sperm living in her spermatheca. And now I come from a behavioral ecology background where we learn that mating can be extremely dangerous and keeping sperm alive inside your body can be extremely costly. And so I've always wondered, why are these females mating and storing sperm if they're not going to use it? And so this is why I wanted a genome. And one of the first things we did was look for genetic differences between these two strategies. So we did whole genome resequencing of 9 solitary females and 9 queens. Now this is a relatively limited sample size for this type of analysis. We used FST as a measure of genetic differentiation. There are a lot of reasons why we had a limited sample size. One is that these are sort of the mammals of the bee world. They're incredibly difficult to work with, especially if you want to really understand their entire life history in terms of social terms. So we took a relatively conservative approach given this limited sample size. We calculated FST based on a moving average across windows consisting of 45 SNPs each. Despite not finding any fixed differences in those windows and a relatively low average FST across the genome, we did identify 26 differentiated regions in the genome. And then we looked within those regions and found 444 SNPs that we consider to be significant outliers. They have FST greater than 0.2, which corresponds to roughly a 95% confidence interval. And what you can see, this is the location of all of those outlier SNPs, and the vast majority of them are in non-coding regions. And so we wanted to understand what effect they may be having on the biology of these females. So we interrogated the outlier SNPs in potential regulatory regions, so the promoters and the introns. And we found that 32% of these outlier SNPs have a significant effect on the probability that one or more transcription factors will bind in that potential regulatory region. So what I'm showing you here is just an example of one of that general pattern. And so we have here a gene model. This happens to be a lipase with potentially vitellogenic functions. And then located here are the sites of the outlier SNPs. And the color of that circle corresponds to the transcription factor that could potentially bind there. And here I'm showing you the motif, the binding motif for those individual transcription factors. So here we have the allele that's most probable in the solitary female and the allele that's most probable in the social female. And the box indicates where that transcription factor is most likely to occur. So as an example here, this is trithorax-like, which is a highly general transcription factor. I've just picked out one of its many functions that could potentially be related to this difference in the social biology of these solitary and social females, to show you that this transcription factor is more likely to bind in the upstream region of this particular gene in a social female than in a solitary female. Other examples in this particular gene include abnormal chemosensory jump 6, which regulates gene expression of odorant receptors and development of olfactory neurons. And then deformed, which is, of course, a classic Hox gene involved in head and brain development. So these are just examples, but it sort of illustrates this general pattern that we found, which is that regulatory control of genes related to development seem to be underpinning social plasticity in this species. And this is really the hypothesis that we set out to test with this genome. And we were able to do so by looking at, by using 2 transcriptome data sets that Burrell Jones had generated a couple years prior. And that includes one that gives us clues about expression plasticity during development. So transcriptomes from eggs, larvae, pupae, and adults across both sexes. And then also a data set that gives us information about expression plasticity between social phenotypes, and that includes queens, workers, and solitary females, and then what we call replacement queens in abdominal tissue and brain tissue. So we were able to use these data sets and our new genome to explore the relationship between developmental plasticity and social plasticity. And we did this, we found several lines of evidence that support the hypothesis that these two are interrelated. I'm only just going to give you a snapshot of one of those. But basically, what you're looking at here are these scatter plots where every single point is a different gene. And on the x-axis, we have the degree to which expression of that gene is dynamic across development in terms of between the sexes. And then on the y-axis, we have the degree to which that gene expression is dynamic across the social phenotypes. And that's for both abdominal and brain tissue. And so what you can see is that these are generally positive related. So genes that are differentially expressed to a large degree between sexes are also differentially expressed to a large degree across different social types. And this holds true when we look across developmental stages as well. So eggs, larvae, pupae, and adults. And in fact, when we took this and we asked, okay, well, what are the best predictors of social plasticity between our different social phenotypes in terms of gene expression? We found through model selection methods that developmental plasticity is among the strongest predictors. So the degree to which a gene is expressed dynamically across development predicts how much it's differentially expressed between our social phenotypes. So I just want to leave you, that was one line of support for this, this general pattern that we're finding between a relationship between developmental plasticity and social plasticity. And we tend to, when we think of social insects, think about developmental plasticity. It's one of the most celebrated features of life in a social colony, where we get different morphological and behavioral phenotypes emerging from very similar genomes. But what our analyses of the Megalopta genome suggests is that this could also be potentially a driver of social evolution, where changes, the genes that are functioning during development are providing the scaffolding from which social plasticity emerges. And with that, like I've alluded to, these results have all just been recently published, some as recent as a week ago. So I'd encourage you to check out those papers if you're interested in the full story. Thanks.
Thank you very much, Karen. That's really cool stuff. Time for one very quick question, because you're almost on time. So it's fascinating to think that developmental and social plasticity are linked. Are the genes that are differentially expressed in both contexts members of any well-known signaling pathways, or are there real surprises?
Yeah, so that's a tough question to answer, because so many of the genes that are linked in both cases that are linked to social plasticity are also linked to developmental plasticity. And so it just sort of becomes it's almost all the genes, right? So it's hard to point to one particular sort of molecular function or biological process. We tried to not rely on gene ontology analysis too much, but in general, yes, there's a lot of regulatory genes, transcription factors, signaling genes, things like that tend to come up over and over for sure.
Yeah, and I think there'll be some follow-up questions on regulatory genomics and how to deal with that. But as we're exactly on time, for now, we'll leave that for the end. And Kristen, you're welcome to introduce the next speaker. Thank you, Karen.
Thank you so much. And moving right along, we're going to go from masses of different genes to contemplation of a supergene. Yannick Wurm, we're looking forward to your talk, and by all means, jump right into it, looking at fire ants.
Can you hear me? Yeah, okay.
Yes.
That's nodding. Thanks so much for having me, and then for putting together the conference in this format that really works very well. So I'm really fascinated by the huge diversity of social lifestyles that we see across the ants. And many have studied these for a long time, and so we know a lot about many different aspects of ant life, including there's big theoretical frameworks that are very robust to understand the trade-offs involved in social evolution and altruism and social lifestyles. But compared to that, we know, we're only beginning to understand what types of genetic changes can underpin changes in social lifestyle. And to add a different way of thinking about it, if we look at differences between species, well, most of those are going to be genetic, right? But surely they will have started as variation within species. But normally when we think of variation within species, it's not that clear cut. Is it environmental? Is it genetic? So here to shed a bit of light on this, we're going to talk about fire ants. And this species is a worldwide pest, originally from South America, now found worldwide. And what of their characteristics that gives them their name is that they're very aggressive, and when they sting, it hurts, and there's many of them, so it burns for a while. Because of this, people have been trying to eliminate them, and they're the best studied of all the ants. Now, what fascinates me about this species is that here within one species, we have two different types of social lifestyle. We have the ancestral single queen type of social organization and the derived multiple queen type of social organization. And in different environments, one social form does better than the other one. And the truth universally acknowledged for a very long time was that environmental factors determine whether a colony will have one or multiple queens. We'll see it's not completely true. So a while back, we sequenced this guy's genome, and then we did a type, I'm skimming some details, we did a type of genome-wide association study where we took individuals from single queen and multiple queen colonies and compared their genomes. And one thing really stood out, this region over here on chromosome 16, it's bigger than 20 megabase, a really big region of the genome containing several hundred genes that doesn't recombine normally between the two social forms. And so this is a supergene region. And to understand how it works to determine social forms, I'll take you back to prehistoric times when a bunch of behavioral experiments were done based on a single marker gene. So in a single queen colony, everyone is big B, big B. If a colony has individuals with the small b version of the supergene, then that colony will accept multiple queens, and all the queens must be heterozygotes, big B, small b. Now, small b individuals, they're kind of like recessive lethal. And this system is fixed. So, if you add another queen to a single queen colony, she's going to be executed by the workers. Same thing happens if you put a big B queen into a multiple queen colony. But a heterozygous queen, she smells different and she'll be accepted into the nest. This has been described as a selfish or a greenbeard system because the workers that have small b kill the queens that do not have small b. Here, it's a supergene region, right? And such regions, they are thought to evolve when alleles at two different genes work together well in a certain combination. Now, the most widely studied supergene regions are sex chromosomes, but there are many others across a variety of species where such non-recombining regions control major phenotypes. Here, this small b supergene is responsible for making the colony into a multiple queen colony. So one of the pressures it will face is that selection will try to make it better and better for multiple queen colonies. But then there's another pressure too that comes from the fact that it's alone. It cannot recombine. And so like a Y chromosome, we expect it to degenerate. It turns out that our system is really young, and that makes it actually a really good model for understanding supergene evolution in general. And then furthermore, as indicated by others, male ants are haploid, so we can get really precise genotypes very easily, despite the supergene system being so young. And so I want to talk about two things. First, kind of the evolution of genome structure, and then also the evolution of gene expression. So Eckhard Stolle did some bionano long read mapping. And so that means he constructed something called optical chromosomes from big B individuals, so haploid males, and independently from small b individuals. And here, when comparing the different assemblies, it turns out that there's at least 3 major inversions. So bits of the genome where the sequence is similar, but it's flipped around the wrong way in small b compared to big B. And it's easy to understand how that can mess up meiosis, or it can mess up recombination. It can prevent recombination from occurring normally. But another pattern also stood out, and that's this one that you see here, that among the small b individuals, there seemed to be an excessive sequence, and there was no similar excessive sequence in big B individuals. We looked at this using an independent approach, genome size estimation from Illumina data, and it held, and it's suggesting that the supergene region, the small b version, is almost twice as big as the equivalent region in big B. So what's going on here? You know, in a sex chromosome thing here for humans, the Y is really degenerate and really small. And we're saying small b, it's a bit like a Y chromosome, but actually here it's doubled in size. So because of the features of this system, we've been able to pinpoint a stage in supergene evolution that is likely common, but is rarely seen. In this kind of context where recombination ends up being completely suppressed, well, there's still useful genes in there, but getting rid of them is too lethal. And so instead we're seeing the first phase here where mildly deleterious mutations are accumulating, repetitive elements, and the region is growing and growing and growing. And during that time, the genes are degrading slowly. As they're degrading, the rest of the genome has time to begin to compensate for this using multiple mechanisms. And then once that is in place, well, the cost of losing genes in that region is lower, and you can end up with what we see with old sex chromosome systems. Shifting gears, Carlos compared gene expression between single and multiple queen colonies, but also between the 2 possible alleles within the heterozygous individuals that exist only in multiple queen colonies. And this, you know, these 2 types of comparisons identified a lot of differences. And at first look, it's tempting to conclude that such differences are signatures of adaptation. But when we looked at it and thought about it quite a bit more, we realized actually it's more complex than that. So for most genes, we see lower expression of the small b allele. And that's actually what you expect, given that small b is degenerating. And more of that occurs if there's more amino acid level degradation of a gene. However, despite that, for most of these genes, they're expressed at a similar level in both social forms. And so that indicates that gene-specific dosage compensation is taking place. For a small number of genes, we see higher expression of small b alleles. And for these ones, this leads to higher expression in multiple queen colonies. So this is really the type of signature we expect from genes that are evolving to adapt to the social form. However, even among those genes, we don't see much fine-tuning across tissues. So selection is probably shaping this at one level at the cost of misexpression in other tissues. So take-home message here is that we have to be really careful with this kind of system when examining or interpreting the expression patterns. Okay, so we have this weird system in the fire ants. Is that the exception or the rule? With some Swiss colleagues, we looked at a completely different system where the two social forms independently came to be, and we basically applied the same approach. And it turns out that in this species, chromosome 3 is this type of large supergene, or social chromosome, as they're sometimes called. So nature found the same type of genetic architecture to have two social forms in this independent system. But it turns out that region is not orthologous to the one that we have in fire ants. This makes you think, okay, maybe this kind of thing is needed to have two social forms in the species, or for other variation in social organization, or for other major innovations. And I think it's partially true. I'm gonna sidestep and mention something completely different. I mean, you guys are all genomicists, and so you know this as well as I do. The way medicine has changed over the past 10, 15 years for research and for diagnostics of patients is just tremendous. And we're all aware of some of the challenges that face many invertebrates outside. And here I think there's a strong opportunity, and we're beginning to grasp this opportunity, to use those same genomic molecular diagnostics approaches to understand the health of wild pollinator species, and then also to help improve the regulatory mechanisms that aim to control the things that we as humans do to affect the environment. And so we have another line of work that I'm not going to go into further detail about now. To summarize overall, we have this system in the fire ants where small b supergene determines social organization. It's a young system, and so it's actually a really good model to understand supergenes in general. And we uncover many patterns, many of which are signatures of degeneration of the supergene, and some of which are very likely to be adaptive. Thank you very much for your attention.
Thanks very much, Yannick, and for keeping on time. So that means you get some questions directly. We have a question from Rich Meisel, which he asked, in case you don't get to it, is there a fitness cost to the B haploid males? And you didn't get to that, did you?
Correct, I did not get to it.
Okay.
Yeah, so, the male stage is indeed in this system you would expect it to provide strong purifying selection on the small b. Here, despite that, we see that indeed there is a fitness cost. So they have lower sperm counts. Often when a queen mates with a small b male, she's going to mate again, although that normally doesn't happen in this genus. So there is a fitness cost.
Okay. It wasn't necessarily clear, maybe, from the cartoons that you put on the resolution that you have in terms of what's actually in big B and little b. So is the gene content the same? Are there differences? Are the same genes there, same copy numbers there? Are there losses? Do you have that level of resolution?
Partially. So if you look at a really superficial level, the gene content is the same, with 1% or 2%. So there's about 400 genes. There's a few that you only find in one place or the other, but that's similar to any kind of two haplotypes you would look at. It looks, however, like there's more variation among small b copies than we would think. So most of the analyses have been done on an invasive population in the United States or in Taiwan. If we go to South America, we actually see much more diversity among both big B haplotypes and small b haplotypes. And because a lot of the evolution of small b seems to be stuff being rearranged and jumping in, etc., we actually need to consider that much better than has been possible so far.
Yeah. And finally, one quick one from Marion Goldsmith. Any thoughts on why fire ants keep holding on to this two social systems through time?
So the way this genetic system works right now is that the multiple queen system cannot go to fixation, even if single queen colonies were useless. And it's because the multiple queen colonies always contain the genetic material for making single queen colonies. And so we cannot have loss of the single queen colonies right now. I think that could happen if there were another major rearrangement or a chromosomal fusion or something like that. But yeah, so that's one of the reasons. The other reason may be that kind of this balancing selection where the environments in which these species are keeps changing back and forth. In the native range, we often find them in disturbed habitats close to river flood areas where the environment does change dramatically very regularly. And so having those two things can be an advantage too.
Okay, thank you. I think it's time to move on. So if you can stop sharing and then Kristen can go ahead with introductions. Thank you, Yannick.
Yeah,
Okay, thank you so much. And building on some of those themes about what's stable as multiple systems in the population and that you can't breed to a single alternative, now we're going to switch out to a crustacean, but think about experimental evolution and potential for adaptation. So David Stern, when you're ready, go ahead and share.
Okay. All right, thank you very much. My name is David Stern. I'm a postdoc in the Lee Lab at the University of Wisconsin-Madison, and I'm going to talk a bit today about a project I've been working on using experimental evolution and population genomics to understand the genomic basis of adaptation in the Baltic Sea in copepod Eurytemera affinis. So the Baltic Sea, like many other coastal and brackish water habitats, is predicted to undergo rapid and serious declines in salinity in upcoming decades due to increased precipitation and ice melt in the northern latitudes. And aquatic species typically have narrow ranges of salinity tolerance, so this may have a large effect on these ecosystems, but we don't really understand how populations are going to respond to this rapid change in salinity. So in order to do that, we really need to understand the genomic architecture of salinity adaptation. And the reason for that is the qualities of the genomic architecture of an adaptive trait will help determine both the probability of adaptation for a population, as well as our ability to predict underlying genomic responses. For example, as the number of loci contributing to adaptive variation increases, theory predicts smaller individual effects of each locus, subtle and small frequency shifts once responding to a new selective pressure, less genomic parallelism and more heterogeneity as different populations are exposed to the same selective pressure, and more, but the retention of more standing variation as we don't expect large shifts to fixation. Of course, we don't know where salinity falls on this, salinity tolerance falls on this continuum, and more broadly, we don't know when these predictions hold and when they do not. However, detecting targets of selection becomes more difficult as a trait becomes more polygenic due to these small, subtle frequency shifts, and as such, we expect for salinity tolerance as a complex trait. So to deal with this, we decided to take a laboratory natural selection approach using as a system the copepod Eurytemera affinis, which is not only a major supporter of ecosystems in the Baltic Sea and other coastal ecosystems, but as a laboratory model, it has short generation times, around 20 days, and it can be maintained in large numbers in the lab. We have a reference genome. So what we did was expose multiple replicate populations to one selection pressure, in this case salinity decline, over 10 generations. We had 10 replicate treatment lines, all derived from the same site in the Baltic Sea, that were exposed to declining salinity for those 10 generations, and 4 control lines that were maintained at the same salinity throughout the duration of the experiment. And then we performed pooled whole genome sequencing at 3 time points, generation 0, 6, and 10, to track SNP frequency changes over time. And by comparing the frequency trajectories of the control and the treatment lines, we could detect signatures of natural selection to declining salinity. So we want to detect loci with those signatures of selection, assess the number, genomic distribution, and effects of those loci, and assess the degree of repeatability at the genomic level for those replicate lines, and ultimately use this information to evaluate wild populations for their adaptive potential. Although today I'm just going to talk about the first 3 points. And in the interest of arthropod genomics, I just wanted to mention this one challenge we came across where our reference genome generated as part of the I5K project in 2013 was actually derived from a subclade of the species from North America that we found to be too genetically divergent for our sequence reads to map reliably. Only around 30% could map. And I think this highlights some of the challenges of genome sequencing efforts for species complexes and, in general, poorly described a species diversity that's quite common in invertebrates. Recently, we've been trying to assemble new reference genomes for this and other subclades using long-read PacBio and Oxford Nanopore data and long-range Hi-C data. One problem is that our Oxford Nanopore runs are failing due to some unknown contaminant clogging the pores. So if anybody listening out there has come across a similar problem in some kind of similar critter, I'd be really excited to talk to you about how you might have overcome that problem. But what we ended up doing is using an Illumina transcriptome from this population, which is easy to assemble, in order to guide the assembly of our pool-seq data into non-coding regions of the genome, in order to then capture that variation at those potentially important sites in the genome. So to highlight some of the results, we found overall a moderately polygenic response to selection with around 1,000 SNPs responding to this selective pressure. Overall, they experienced relatively subtle frequency shifts. As you can see in the histogram on the left, in purple is this frequency shifts from the starting population at generation 6, where there's an average of 5% change and an average of 10% change at generation 10. We don't see many large sweeps to fixation. We do find relatively large selection coefficients. The histogram on the right shows the distribution of those selection coefficients in purple for the experimented selected loci, which tended to be relatively large compared to similar studies. And overall, there's a small number of somewhat large effect loci, but a larger number of smaller effect loci exhibiting subtle frequency changes, pointing to a moderately polygenic response. We found a surprising level of parallelism and repeatability across the replicate lines, considering those 1,000 or so SNPs with significant signatures of selection. In this PCA plot on the left, the 2 red dots are the 2 samples from our starting population. In yellow are the control lines, and in purple are the treatment lines. And you could see not only a clear deviation from the control lines by generation 6 and 10, but also a clustering in this space, suggesting a convergence of SNP frequencies in these replicate lines. Another way to look at this is the plot on the right, where I have pairwise overlap in significant SNPs between replicate lines. And in purple is our real data. You could see moving from generation 6 to generation 10, there's a marked increase in this parallelism or the overlap in significant SNPs as selection went over time. And we also simulated selective sweeps to get an idea of what a baseline expectation is. We found that our real data exhibited even more genomic parallelism than expected from simulations, suggesting that something particularly interesting is going on with this trait, and a general rejection of our expectations from a polygenetic model. And then finally, we found that selection tended to act on SNPs starting at intermediate frequencies. The histogram on the left shows the frequency distribution for non-selected SNPs in yellow and experimental selected SNPs in purple. You can see a clear shift towards higher frequency alleles in the experimented selected loci. And since our tests were not actually biased towards the detection of those high frequency alleles, we think that this is a signature of balancing selection, maintaining this variation, which is in agreement with previous work suggesting that balancing selection could promote adaptation to novel habitats by maintaining important genetic variation at high and intermediate frequencies. So just to sum up, we found that salinity adaptation appears to be moderately polygenic with around 1,000 contributing loci of various varying fitness effects. Polygenic adaptation can indeed be repeatable despite theoretical expectations, and putatively adaptive genetic variation might be maintained by balancing selection in the wild. And I think all of these points point to a high probability of successful adaptation in our copepod, which is a good thing. And I'd just like to thank my lab, the Lee Lab, our collaborators, and funding sources. Thank you.
Thanks very much, David. We have 1 minute, so perhaps you could enlighten us on the genes and gene functions that you find to be under selection. Given that the crustaceous species sampling in terms of genomics is still, only just getting going, and crustaceans are so far evolutionarily related to other arthropods, how do you go about actually functionally annotating these genomes to then kind of make these conclusions?
Yeah, that is a challenge due to the divergence from well annotated species. Our lab has actually led some efforts to really well annotate ion transporter genes based on phylogenetic analyses, and we found consistently that a number of ion transporter genes seem to be under selection in this system, which makes sense given their clear and important function in salinity tolerance. We've also found gene regulation to be a enriched gene function. And the fact that we see the same sets of genes, the same paralogs across different studies of different subclades from this group suggests, again, some limited path to adaptation in this system.
Okay, so you go for what you know first, and then hopefully
exactly.
in the future you'll be able to figure out what all the unknowns are, right?
One day.
Okay, thank you very much. We'll save the rest for the 15 minutes at the end. Thank you, David.
Okay, then I'm happy to introduce the next speaker. If you were with us in the previous Eco-Evo session, there are a couple of foreshadowings where we saw the tremendous eyes in the mayfly, and we also already had a talk about Drosophila. Next up, we have Nico Posnien looking at eye size variation in Drosophila.
So, can you hear me now?
Yep.
Okay, great. And you see the slides?
Yep.
Perfect. Okay, then thank you very much. So my group is mostly interested in contributing some answers to kind of questions related to the molecular basis of morphological diversity observed in nature. And as a model, we study morphological evolution in Drosophila, which is kind of funnily the only critter on this beautiful picture that gets stabbed in the face by this bear. So I don't know if that was done on purpose or not. Specifically, we are studying variation in head morphology, which has recently been shown to be highly diverse in Drosophila. And we started a few years ago to focus on a few species of the Drosophila melanogaster group and a few species of the Drosophila virilis group. And today I will share some data on these two species from the virilis group. Drosophila americana is present in large parts of the eastern US, while Nova mexicana is kind of restricted to the southwest. And so to study morphological evolution, we follow this kind of rationale here. We assume that morphological diversification is a result of variation in embryonic and post-embryonic development, and that these changes are somehow coded in the respective genomes. So therefore, the first step is always a thorough morphological comparison. And these are two representative heads of the two species. And we can actually quantify morphological differences, for instance, using geometric morphometrics. And in this case, we show major differences in the head shape. And this data suggests that we see differences in compound eye size, which we then, by measuring manually the area of the eyes, could confirm kind of quantitatively that there's major difference in eye size. So therefore, we can conclude that eye size and head shape are highly variable between Drosophila americana and Drosophila nova mexicana. To establish the genetic basis of these observed differences, we first crossed the two species and generated F1 hybrids. And since those hybrids showed intermediate head shapes as well as intermediate eye sizes, we went on to actually backcross these F1 females to Nova Mexicana males. We got like more than 500 recombinant offspring, which we then phenotyped, and we genotyped with kind of old school indel markers to keep track of individual chromosomes. And if we we can group these more than 500 individuals based on the presence of Drosophila americana chromosomes in the Nova Mexicana background. And we see that an increase in eye size the more Americana chromosomes we add to the Nova Mexicana background. So this data suggests that variants on different chromosomes contribute additively to eye size differences in these two species. To increase the mapping resolution beyond chromosomes, we set up sister-brother matings for 17 generations, and we obtained about 160 recombinant females, which we phenotyped, and then we grouped these individuals into 4 quartiles, with quartile 1 containing about 40 flies with the smallest eyes, and quartile 4 about 40 flies with the biggest eyes. We extracted genomic DNA from each of these quartiles or from these pools, and we sequenced 100 base pair paired end reads, which we mapped to the reference Drosophila americana genome, and we estimated allele frequencies and performed a GWAS analysis. Yes, and if we compare, for instance, quartile 1 and quartile 2, so the extreme quartiles, extreme phenotypes, we end up with this crazy Manhattan plot. So superficially, we found large chunks of most chromosomes contain variants associated with eye size differences. So with that, this GWAS approach kind of confirmed our backcross results that variants on most chromosomes contribute to eye size variation between Drosophila americana and Drosophila Nova Mexicana. But the big question remains, why do we see these highly structured genomes? And to answer this question, we first improved existing highly fractured assemblies for both of these species. And actually we achieved that by adding MySeq data, kind of still a little bit old school, but that allowed us to link previously unlinked contigs. And based on these improved genomes, we were able to map the exact breakpoints of most chromosomal inversions that have been shown to exist in this species complex. And as shown here, each chromosome carries at least one large inversion that is fixed between Americana and Nova Mexicana. And if we roughly map these, the locations of these major inversions on this Manhattan plot, and you see that variants associated with eye size differences are very well correlated with the presence of inversion. So this data therefore suggests that causative variants are linked to chromosomal inversions. So that is great, but of course that doesn't bring us any closer to potentially meaningful candidate genes or candidate SNPs. So we asked what other information could we integrate to reveal interesting candidates. And it turned out that the integration of phylogenetic information as well as population genetics data for Drosophila americana was quite powerful. So specifically, it has been suggested that Drosophila Nova Mexicana actually derived from a northern population of Drosophila americana. And interestingly, we found that eye size is highly variable between northern and southern strains of Americana, with Drosophila americana having smaller eyes compared to northern strains. Or yeah, northern strains have smaller eyes. So therefore, we actually generated pool-seq data for Drosophila americana populations from the north and the south of the species distribution. And using this data, we revealed various genomic regions that show genetic differentiation between these populations. And now, in the light of the intraspecific variation in eye size, we assume that variation that shows that variants, SNPs that show allele frequency differences between northern and southern populations of Americana, and at the same time can explain interspecific differences in eye size between Americana and Nova Mexicana. These are prime candidates to actually look at a little bit closer. So concentrating on chromosome 4 and 5, we actually managed to identify a few SNPs that are shown in green here that match these criteria. And interestingly, among those SNPs, we found a subset of significant SNPs located inside or nearby candidate genes that are known to be involved in eye development. And as many studies already shown for other traits, many of these SNPs are located in putative regulatory sequences such as introns. So therefore, the approach of integrating population genetics data allowed us to narrow down putative interesting SNPs. And we could show that some of these SNPs are potentially associated with differences in the regulation of genes involved in eye development. And with that last final bit of data, I would like to finish thanking my postdoc, Michael, who actually headed this project, as well as our main collaborator on that project, George Vieira. And well, if you're interested in that project, I have a postdoc position to actually follow that up with RNA-seq and ATAC-seq data analysis, so integrating gene expression and gene regulation data. So if you're interested, just drop me a message. And with that, I would like to thank the organizers. So it's a really great organization, great conference, and thanks everyone for listening. Thank you.
Thank you, Nico. Exactly on time, but used up all your question time for direct questions. So we will keep those for the very end, if you don't mind, and move swiftly on. Thank you.
Thank you.
Okay, so moving right along, we're going to shift from one very prominent visible trait of head shape and eye size to looking at bumblebees and color variation. Sarthok Rahman, we're looking forward to your talk.
All right, can you hear me? And can you see the slides, please?
Looks great.
Yep, all good.
Okay, all right, cool. So hi everyone, my name is Sarthok and I'm a grad student from Heinz Lab, Penn State. So today I'm going to talk about the genomic basis of mimetic color variation in bumblebees, or as I often simplify this into, how did bumblebees get their colors? So bumblebees are exceptionally color diverse. As you can see, there are around 260 species around the world, showing more than 400 color patterns. As we can see that the color comes from the hair, and this exceptional color diversity is linked to aposematism, as they're the part of the extensive Mullerian mimicry complexes across the globe. So how did these mimicry rings contribute to phenotypic diversity? So in one geographic region, as you can see, that several species can converge into a similar phenotype, and also you can see that one species can have different color forms in two different geographic regions. So this makes actually bumblebees a great system to study natural selection in action, because this gives us multiple natural replicates to understand the evolutionary genetic processes. So we are using this new model system to dive into genomic basis of mimetic color variation, and we start by focusing on a specific mimetic species right here, Bombus melanopagus, which shows a red versus black switch in their adrenal pile, along with its co-mimics. And as you can see that in they are the part of the Western North American mimicry complex. In Rocky Mountain region, we see the red is the most dominant type, where in Pacific coastal region, the black is the most dominant form. So we were able to test the genetic basis of this color variation in Bombus melanopagus using sequencing of natural variants. as there's two dimorphic forms. They actually meet in the hybrid zone, and their population is completely admixed. And we are pretty lucky that we went with a pretty low sample size, 10 black individuals and 11 red males. And we used them for a GWAS-based approach, a pretty standard GWAS-based pipeline, sequenced Illumina high-sec reads, aligned them to a closely related bombus impatiens genome, which was available then, and variant calling and genotype-phenotype association to find out the color locus. So as you can see, from the Manhattan plot in the bottom, there's only a single association peak which actually lies between two genes, abdominal A and abdominal B. So we heard about the Hox genes today a lot, and this is an awesome find because these are the two major genes driving two Hox genes actually driving segmental identity. And this is pretty evolutionary unexpected because as you can see that their spatiotemporal expression pattern across all arthropods are very conserved. So now we have more questions to answer. Yes, we got the color locus, but what's the gene driving this coloration? Is it abdominal A or abdominal B? So to figure out that, we had to develop an extensive staging protocol. So where we actually had to observe the pigmentation process happening right across all developmental stages of bumblebees. And then by doing qPCR of bithorax complex genes, which is abdominal A, abdominal B, and UBX across those developmental stages, we actually figured out it is actually indeed abdominal B, where we see that right before the adult eclosion, in the 0-hour callow stage, based on our staging protocol, we see a 10 to 20x upregulation in the red form, but not in the black form. So this is also a case of homeotic shift where the typical expression of abdominal B is shifted to more anterior parts of the abdomen to drive the phenotypic change. So the next question for us is how this upstream connector abdominal B, a Hox gene, is changing the pigmentation between the color morphs, okay? So this is, before that, all I discussed is all published and all the data is deposited in NCBI, all codes in Dryad, so you can have a look or we can discuss later. But the new part that now we are trying to figure out what are the likely downstream targets of abdominal B. And thanks to the previous research from our lab, that we know that the red versus black switch is actually a shift of portion of eumelanin to pheomelanin. So we can obviously assume that, okay, the melanin pigmentation pathway is probably being implicated here. So we are very lucky to know quite a bit about the Drosophila melanin pathway. And there's a vast amount of research from Drosophila, which basically shows us that practically every melanin gene can be modified in cis to generate melanin color variation in Drosophila. And obviously those genes like pale, ebony, and tan, those are really good, excellent candidates for bumblebees too. But what's actually happening in bumblebees? That's our question. So we try to figure out those downstream genes in the melanin pathway of bumblebees, and we took an RNA-seq approach. And thanks again to our very detailed staging protocol, that we've been able to select the right stage. As you can remember that, as we said, that 0-hour callow stage where this all magic's happening. So we sequenced 6 replicates each for transcriptomic sequencing, Illumina parent sequencing data, standard RNA-seq pipeline for using Callisto and DSEQ2, and we used the bombus impatiens annotation and obviously blasted against the Drosophila to find out the homologs. And ultimately, we figured out 307 differentially expressed genes between two color morphs. Okay, so now when we start looking at the most differentially expressed gene list and try to put in the context of Drosophila pigmentation melanin pathway, that for both black and red forms, we revealed the top highly expressed genes are actually the major melanin pathway candidate genes we assumed before. So for example, you can see that ebony is highly expressed in the red form. And then for the black form, we see the pale and peroxidase. All of those are either part of the melanin pathway or the precursors to it. And another really interesting find is the nubin here, that which could be a potential mediator gene between the upstream regulator abdominal B and the downstream pigment genes. And we are currently doing the qPCR using the same staging protocol to figure out that is how the abdominal bee has the connections between these downstream regulatory genes across all the developmental stages, looking at their expression pattern. So this is, as I noticed that many of us are working on a new model system. So I just wanted to summarize that what are the major takeaways from our research. So we had to take a genomics heavy approach because functional validation is pretty hard in Hymenoptera. We are not as lucky as Lepidoptera or Coleoptera scientists to do a CRISPR or RNAi as easily as it could be done in those systems. So we had to take a GWAS and followed by qPCR and RNA-seq approach. And this basically gave us a lot of information. We were able to connect the upstream and downstream players. So it worked pretty well, I would say. And then obviously we had to deal with a budget limitation where as Karen and Yannick said that everyone's going for the haploid males, we also did that. And that was a pretty good, efficient sampling strategy for GWAS. And I would also like to take an opportunity to thank everyone who is providing this amazing community resources. And we couldn't do anything without those ones. And also every time there's a new bumblebee genome comes, it actually helps us to expand our research. For example, we plan to do the comparative genomics part of this project by looking at the evolution of Hox genes and pigmentation genes across the genomes using the new 17 genomes just came up in bioRxiv from Professor Chen Sung and Robert Waterhouse group. So we are looking forward for that collaboration. So I would probably finish right here, but before that, I want to thank all of the current and former members of Heinz Lab, and also our funding sources and our collaborators. And also I'd like to give a big thank you to the organizers of this symposium for giving me an opportunity. And so far it's great. Probably everyone would just like to do the Zoom meetings in next year's, I guess. So I'll just finish right here. So I'll invite questions from you in Q&A, and I'll be available right after this session for discussions in the Slack space.
Thank you, Sarthok.
Thank you.
I think we would like to move straight to our last speaker because it's a very short speaker, not short speaker, sorry, apologies, a short slot. And then we can have the discussion, the full discussion at the end. Okay, thank you.
Thanks.
So just as a quick comment, the questions definitely are coming in to look forward to. And just to let you know, we've peaked at over 300 participants in this session, so there should be indeed lively Q&A at the end. But without further ado, Emily Jennings is going to tell us about a cockroach.
Hi, everybody. Can you hear me all right? See the slides? Cool.
Yep.
So thank you so much for the opportunity to speak. Bear with me in my super short time slot. Somebody, Josh, decided that I was going to present my entire PhD dissertation in 5 minutes. So this should be a real adventure. Without further ado, I'm going to tell you about a multi-omic examination of reproduction in a really unique little cockroach. First off, I just want to say a little bit about live birth in eukaryotes overall. It represents one of the largest instances of convergent evolution among eukaryotic life in both vertebrates and invertebrates, evolving independently at least 60 times in 22 families of flies alone. But like we mentioned before, we study this in a cockroach, Diploptera punctata, also known as the Pacific beetle mimic cockroach. The specific flavor of live birth that this guy uses is that it has a pseudouterus / pseudoplacenta, which is called a brood sac. It nourishes 9 to 12 embryos over the course of 70 days of pregnancy by providing them with a milky secretion rich in proteins, carbohydrates, all of the good stuff that you want to grow. And here's a micro CT scan of a pregnant female and a micrograph of their brood sac. We're going to talk about a transcriptome analysis we did, a microbiome analysis, and then a bacterial symbiont genome sequence very, very quickly. And this is the overview of all of our methods. And I know there's a lot going on here, but we used Illumina HiSeq to do our RNA-seq. We used pretty standard 16S ribosomal RNA amplification, amplicon sequencing to do our microbiome. And then again, we used Illumina HiSeq to do our bacterial genome, where we kind of took a pseudo-metagenomic approach because it's really hard to get that separated from the host. High-level overview of our transcriptome de novo assembly is we had around 12,000 extracted coding sequences, and a really lovely set of BUSCO scores where we got the overwhelming majority of BUSCOs either as a complete single copy, a complete duplicate, duplicated copy, and a handful of fragmented. So this was essentially one of the largest genetic analysis of this species thus far. When I started this, there were like 70 total sequences in NCBI for this. So this was something we were hoping would be useful broadly. As far as functional analysis, basically we found that there were distinct expression changes correlated with each stage of pregnancy, but the bulk of these enriched proteins were highly related to the milk secretion and overall in the whole body, as well as specifically in that brood sac. Looking at gene ontology, the largest groups that were enriched are cytoplasmic translation and ribosomal biogenesis, which if you're going to pump out a whole bunch of proteins for milk, that makes a lot of sense. Looking at the microbiome, we looked at embryos, newborns, the entire suite of juvenile instars all the way to adult females. And essentially we saw that in our embryos as well as newborn cockroaches, there's like nothing except for a single bacteria, Bladobacterium, which is unique to cockroaches, and the only known vertically transmitted bacterium. But we were kind of curious if, you know, maybe because they had this extended pregnancy, there would be something else, but it turns out there's not. And it's not transmitted through the birthing process like invertebrates either. But we do see during that first instar, a rapid accumulation of diverse bacterial taxa, and then it just kind of flatlines throughout development. So that first instar is really important in developing the profile. This is a more quantitative representation of that, looking at the Shannon Diversity Index. So you can see here that diversity is super low for both embryos and newborns, jumps up and plateaus throughout the instars. Because that single bacterium, Bladobacterium, is basically the only thing present in our embryos and neonates, we wanted to see if we could look at what it was doing specifically and if it varied from other sequence strains. So we found a genome of around 623,000 base pairs, which is on par with other sequence strains of this, and had, you know, a very standard endosymbiont, non-free living bacterial characterization of that genome assembly. If we look at what it's doing functionally by using like KEGG pathways and predictions for that, we can see that they're able to synthesize almost the entire suite of essential amino acids, including tryptophan and methionine, with the exception methionine a little funky, but that's a longer story and we can talk about it later. These two things are particularly important because these amino acids are deficient in the milk secretion that feeds those embryos through their development, which is crazy because like methionine is really important and also the first amino acid of every protein. And then really quickly, lastly, because we thought these were really interesting and there's not a lot of resources for it, we started working on sequencing the whole genome for it. This is just a very quick summary of that. It's around 3 gigabases long, which is larger than the German cockroach, but about on par for the American cockroach. And this was a joint collaborative effort with Mark Harrison, Eric Bornberg-Bauer, Steve Tobe, and Elizabeth Marshall. And thank you to my lab, very fast, everybody involved in this. And thank you again for the opportunity to speak.
Thank you so much, Emily, for getting through all of that basically on time. That was pretty impressive. I'm going to try and take it all in now. So let's start with you while we've got you, as if you like, on the line.
Yeah.
Given that there's so many independent evolutionary origins of viviparity, are there other lineages that already have sequenced genomes and genomic resources where you can begin to look for commonalities with your cockroach?
Yeah, that's a super great question. Within cockroaches, no, but across the larger insect group, yes. The tsetse fly is a really big example of live birth in insects, and several different species of that fly have their genomes and transcriptomes sequenced. The most common element that we see mostly with like looking at gene expression and hormonal pathways is the suppression of juvenile hormone to maintain pregnancy. And in all of these different species, whether that's tsetse flies or cockroaches, which are very different lineages of insects, we see that treating them with this hormone or an analog causes an abortion of the pregnancy and cessation of milk production. So that's one example of a commonality across all viviparous insects, essentially.
Do you see the same kinds of milk genes?
No,
Okay.
they're very different. In fact, they have different organs that produce them. So in the cockroach I study, it's an epithelial organ, and it's actually a lipocalin. We think it evolved from a lipocalin-like protein. And that's difficult to tell because lipocalins have like major variations and like very teeny tiny pieces that are, you know, consistent across and conserved. On the other hand, the tsetse fly organ is very different, and they originated from a completely different set of proteins. So it's really cool that these are so different and have such different evolutionary origins, but converged on these really similar traits and using the same hormone pathways to control that process.
Yeah, thank you. Moving back up, so Sarthok, there's a question about splicing for you. So did you see any differential alternative splice variants for these genes that are involved with the color differences that you're observing?
Yeah, I think it's a really great question, and we are actually looking into it. I don't know if I can pull my slides, but I showed that the perioxidase gene, and that actually was different in a transcript variants. But obviously, as I said, that we are working with closely related species, and now we are actually assembling the transcripts, the actual red and black forms from the transcriptomics data we have. So we would be able to give a much better answer. But based on impatience, yes, we have seen the splicing differences, but obviously we need to annotate the genes from actual red and black forms. So
Okay.
this is coming up, yeah.
And so along similar lines, but instead of alternative transcripts, the melanization pathway is widely conserved across arthropods because it's such an important function, not just for coloration, but also in immune reactions and things like that. So are the genes that you were using as like your reference genes in Drosophila, are they all conserved in bumblebees? Are they like extra conserved to other kinds of pathways or families, or are they on average?
Okay, so when we aligned it against Bombus references, yes, we didn't see any difference. That is, it not like that the ebony is there and ebony is not in this species, nothing like that. So with Drosophila, yes, actually I had the list of the pigmentation genes and transcript, and when I blasted, yes, there might not be all the transcripts present, but the gene is definitely there. Yeah.
Okay, so you have a basic conservation of the key players.
Yeah. Yeah, I think I just wanted to add a little bit that most of the time it's just the cis regulatory changes in melanin genes driving the coloration. So maybe we wouldn't see anything like the complete loss of the gene, but maybe some other mechanisms involved. Yeah, thanks.
Okay, thank you. For Nico, we have a question here about gene flow. How much of a role do you think gene flow is playing between Drosophila nova mexicana, the northern population, and Drosophila americana, as opposed to founder Americana populations originating from the northern population? I hope I explained that.
Yeah, so if I get the question right, so I think the key point here would be to whether there is still gene flow between Nova Mexicana and Northern Americana populations. So I I'm pretty sure that there is not, so there's no indications of gene flow. And I mean, there are actually the Rocky Mountains that actually, yeah, would block migration or something of the flies. So far we don't find any indication. But it's very tricky because Nova Mexicana, we so far only have strains that are in the lab in stock centers for pretty many years already. And I know from a few people who tried to collect, and so far it was not really successful to find them. So not really sure.
Okay, so from the genetics to the physiology, does eye size change in particular cells or sub-tissues within the eye, or is it size of cell or number of cell or complexity of the eye? What are the physiological changes that you see?
So in that particular, so between Americana and Nova Mexicana, we actually counted the number of facets like externally, and all of the variation that we see in area of the eye can be accounted for by number of ommatidia difference. So it's like 1,000 ommatidia or facets in Nova Mexicana and 1,400, so 400 more facets. That is, of course, in different Drosophila groups and different Drosophila species. We also have some groups where differences are due to the size of individual facets and lenses. So I think that's pretty variable. So it's pretty much a convergent thing that happens, that eye size increase or decreases.
Okay. And then following on to that, from a question from Fringy, maybe philosophical, are these changes the equivalent of flies squinting in the desert? If you gave them some sunglasses, if you gave them sunglasses.
Well, I don't know. I mean, there are reports that Nova Mexicana specifically, there are reports that they may be cave dwelling critters. So that could be, I mean, they also have less pigmentation, so they are they lost a lot of pigmentation in the abdomen compared to Americana. So there may be a cave aspect to it. So they probably don't need sunglasses. But yeah.
Okay, thank you very much. Moving back up the list to David, first of all, there are quite a few people in the audience that are clearly experiencing similar problems. So I hope that you'll be able to arrange to meet with them in the coming days to discuss how people are addressing them. Then we have
that's amazing. Thank you so much.
We have a question. Someone who is curious about the possible role of evolutionary rescue in your experiment. So how were your population sizes maintained, and did you observe mortality?
Yeah, that's a really interesting question. So our populations throughout the experiment, we started off with 10. Actually, 2 of them did not make it to the end of the experiment. They did die. The rest of them, we didn't observe a lot of variation in population size. However, I didn't really do a quantitative analysis of population size changes throughout the experiment. So that's an interesting point and a potential caveat, something I'll think about in the future.
Okay, so you didn't try to cap the population size at any given value?
No, we let them grow to as large as they could. Yeah.
Is that even feasible? I don't know. Like, you have to fish some out, or what?
It's also hard to count them because they're extremely small.
Yeah. So that's where you need some master students to help with your project. Okay, trying to keep on time here. Yannick, we have a question. Oh, okay. Is there any consensus on the evolutionary value of actually sustaining this kind of dual social form?
Yeah, so I skimmed over this. We think there is balancing selection on the 2 social forms. So the single queen social form, the young queens, they're really good at dispersing. They'll do their mating flight at maybe 200 meters altitude, 100 meters altitude. So they can be carried by the winds, and they can disperse, over 100 kilometers when the wind is right, and found their own colony independently. So they're really good at colonizing new habitats. And after there's been flooding or in an invasive range, usually you see that single queen form spreading first. If a habitat is saturated, that's where the multiple queen social form does better. The young queens there, they typically are unable to create their own colony. They do their mating flight at 5 meters altitude and then join an existing nest. If in this kind of habitat, there's a lot of competition, well, a multiple queen colony will outcompete a single queen colony. So I think it's quite clear that the environments where the different social forms do better are very different. And we see them in fluctuating environments. Yeah.
No, that certainly makes sense in this scenario with the dispersal being absolutely important, right? So maybe you mentioned this, but we have a question related to it anyway. So you talked about smelling and killing, basically. So if you don't smell right, you get killed by the workers. So does the social chromosome contain olfactory-related genes that could be like specifically linked to this ability to recognize or not the allele that you're carrying?
Yes, so it's an important thing to think about. Where I always struggle a little bit is that there's more than 400 genes in the region, and if you sample 400 random genes in the genome, it will include some olfactory genes, especially in something like an ant where we have hundreds of them. So if I remember correctly, there's maybe 10 odorant binding proteins in that region. There's maybe 20 or so olfactory receptors in the region. Some of them don't seem to show allelic differences, but maybe a third of them do. And then there's actually some of both that appear to be unique. So where that OBP only exists in the big B form or only exists in the small b form. Those are tempting candidates. Does that prove that they're responsible for something? And I guess you have the same type of thinking about the genes that produce odors. You can have things, there's multiple genes in those pathways. Something there likely contributes to something, but I think a lot more kind of particular disentangling is going to be needed before we can say, okay, this is the culprit.
Yeah, so it's tempting, but you're not going to go there. Okay, thank you very much. Now, moving up the list for Karen, we have a question about whether or not you've begun to explore any epigenetic differences between social and solitary individuals.
The answer is no. We haven't. I've stayed away from epigenetics for the most part, even though a long time ago that was a thing I thought I would do, but I just, haven't done it. So if you're interested in doing it, we have lots of samples in the freezer. So yeah, come on by.
Yeah, I think there's a poster focusing on epigenetics. So there's definitely going to be some discussion around techniques there. Then I have a further question about, given the links that you find between developmental and social plasticity, would you hypothesize that the hub genes that are enabling sexual dimorphisms are also being co-opted for this social plasticity?
Yeah, you would hypothesize that. So some of the classical developmental genes like feminizer and double sex have been found to be differentially expressed between casts in ants and honeybees, and I think stingless bees as well. But we don't really see that in Megalopta. So one limitation is that the double sex gene wasn't annotated in the Megalopta genome, so that is a missing piece of information. Feminizer does have high.
So it wasn't annotated, or it wasn't assembled at all.
Yeah, well, it's not showing up in our annotations, so okay. I'm not exactly sure. I haven't gone in and tried to do any sort of manual annotation with it, but it doesn't show up in our gene set. Okay. Feminizer is there, and it does have high developmental plasticity, as you might expect, but relatively moderate social plasticity in the abdomen, at least.
Okay, cool. So I think I've given at least one question to all speakers. But so there's one last one that I think is maybe a nice broad one for you to try and tackle, Karen. You're not going to get away too quickly. Don't mute just yet. So you clearly showed that regulatory control is very important in these systems. You know, it's not all down to the gene content, but where and when they're turned on in response to what and at what stage of the life. Now, obviously, predicting transcription factor binding sites is notoriously difficult. You know, even in some of our model species where we can get it, you know, pretty good for some of the well-known transcription factors, it's hard. So how did you achieve this, and what precautions did you have to take to ensure accuracy here?
Yeah, so these were all based on sort of motifs that were developed for flies. And then we did, you know, we took a few steps to ensure that at least these transcription factors were found in our genomes. Like, we wanted to make sure that they weren't just fly-specific proteins. And, you know, so we took some steps there, but yeah, otherwise, you know, a lot of this is carryover from predictions from flies, and that's a problem that I think we face in general with social insect genomes. Well, most insect genomes.
Yeah, so I don't know if you noticed, but Mark Halfon is giving a poster on trying to broaden the scope of regulatory elements and motif annotation beyond Drosophila into different insect species. So I think we'll all benefit from that in the long run, definitely. Okay, so Kristen, unless I've forgotten someone or you have a last additional question, we are now 5 minutes over. So I'll leave you to...
Okay, well, no pressure then, but thank you all. I think we've actually done an amazing job across 3 sessions all day long of sticking to time with really great engagement. Just as a quick comment and follow-up, I like how much everyone has emphasized technical challenges and things that come up again and again. So actually, Karen, when you mentioned just now you failed to find feminizer, just from the previous Eco-Evo session, Kim Ferguson found that that was a special gene she had to hunt around for when they were working through the genome. So a lot of the same technical challenges and sometimes related to cool biology keep emerging. I did actually have one question if I can. So building on comments on pigmentation, melanization, and the complexity of those different genes, Emily, you commented that the microbiome shows a major switch when the cuticle is hardening, if I understood. Is that really linked to cuticle, or is it just that living inside a mother and then being thrown out into the wide world is such a completely different environment?
But definitely the latter of the 2 situations. I don't feel like anything very special with regards to the microbiome about like melanization of the cuticle. Rather, it's acquiring the microbiome from the environment. Cockroaches aren't exactly the cleanest things in the world, so they're you know, like they're better than a lot of other things actually, but they're not, you know, the cleanest things in the world. So there's coprophagy that goes on, just hanging out in dirty containers. We feed them dog food, so what they get from their diet.
So quite a
definitely more
environment.
Yeah, environmental acquisition
Okay.
versus something special about that.
Okay.
Just being time in their environment.
Okay, thanks. But indeed, the ecology and the environmental aspects were key things we wanted to pick up across these last 2 sessions. So yes, thank you all. As Rob mentioned, there are some excellent posters. We have over 70. And for those who still have the endurance, because for some of us it is quite late, then there will be a free Zoom chat hangout as opposed to this very formally structured webinar. And thank you so much across the organizers, the speakers, and all of the participants for a great day.
Thank you, everyone. And good night, or have a nice lunch, or whatever you're going to do next.
Questions will appear on the channel as well for ongoing
Exactly.
dialogue.
Thank you, everyone. Bye-bye.